I hope everyone has gotten
vaccinated. I was just going to say it's 6 o'clock Israel time on my clock, or 6:01 to be more specific.
Can we wait for Rivka?
Rivka's here.
Oh, okay.
At least I see her on the list. Okay. Would everybody please open their cameras so that we can see you all and feel like one big community, one big scientific community. So good morning or good afternoon to you all, depending on where in the globe you're located. And I'd like to welcome you to the first in a series of webinars to be held by the Israeli National Academy of Science and Medicine by the Israeli Medical Association. My name is Malki Barrow, and I am the Director of Division of Law and Policy at the Israeli Medical Association, and I've been involved in establishing and helping to smoothly run the Academy since its inception about 1.5 years ago. Just a few housekeeping matters, okay? First of all, it's important that everybody stays muted other than the speaker so that we don't have background noise. There will be time after each speaker for questions and answers. So any questions that you have, please send them by chat. Write them in the chat in English, of course, and I will ask the lecturers to respond to them, as many as we can manage after each lecturer. Okay, at this point, I'd like to get straight to the meat of the meeting, and I'd like to first introduce Ms. Leah Wapner, who is the CEO of the Israeli Medical Association, to say a few words. Just unmute yourself, Leah. Okay.
Okay, so now I can actually speak. Thank you very much. And thank you, Malka. First of all, I'm very, very excited to be here and to have the opportunity to welcome you all and in the name of the Israeli Medical Association. I must admit that seeing everyone here, it really makes me very happy to know that we are part of this important endeavor. I think that we need a special thanks to Professor Rivka Karmi, who it was her vision that actually can see now that is coming to life and is being realized. And I think that it's a great opportunity for scientists in medicine around the world to come together and speak about different issues. And obviously we are now in very challenging times. I think the whole medicine and whole science is being challenged in a way that we didn't see this or didn't know this for many, many years. We know that we have an issue with public reliance on science. We have an issue with trust, and we have an issue with leadership. And what I hope is that this community of scientists that are now taking part of this and will join you will be able to restore some of that trust, will be able to come and voice to the large community of medicine, but also to the public at large, evidence-based topics, evidence-based recommendations, and hopefully we will be able to meet the next challenges that we have in a more based and not ideas. And if you were talking before and I heard the discussion about the vaccination, I just would like to bring that as an issue, as a topic. I think everybody knows that Israel was really rolling out the vaccinations in a wonderful way. We had people going to be vaccinated up and more and above our expectation. But if we see what's happening slowly and slowly, we can see that there are different voices and fiction and fake news creeping in, and even we are facing challenges in this. So if we just take this as an example, I think that it is so important that we have a very sound scientific base for everything we do. That is why it is a great privilege and honor for me to congratulate the Israeli Academy of Medicine. And I really hope that this will be the beginning of many more webinars that will come. So thank you very much. I would also like to take the opportunity to thank Malka. Malka has been really running this organization together with Professor Karmi, and she's doing this after fulfilling a full-time job at the Israeli Medical Association. So thank you very much, Malka.
Thank you, Leah. Okay, now I'd like to ask Professor Rivka Karmi, who is the president of the Israeli National Academy of Science and Medicine, to speak, but she is having some technical difficulties with her camera. So Professor Karmi, Rivka, would you like to speak even without the camera, or should we get back to you later? Okay. Just give it a minute. Perhaps she's getting out and getting in again. Dotan, can you unmute Rivka, please? Or Omer?
I already unmuted.
Can you hear me?
Yes.
Okay, you can hear me, but you cannot see me. I don't think that's a big problem. So thank you very much for all participants here. Let me see. Okay. One more trial. Well, so I'm here. I can see you all. I can admire the fact that there are so many people joining here today. Thank you very much. You've already been told that this is an event hosted and sponsored by the Israeli National Academy of Science and Medicine, by the Israeli Medical Association. This is a newly established academy. It consists of leading physician scientists in Israel, and it aspires to promote high quality, state-of-the-art research by active physicians in the country. Now, if there is a silver lining to the COVID-19 terrible pandemic, it is the flourishing industry of virtual scientific events that make distinguished scientists a little bit more accessible. So it is about our esteemed Professor Benny Deckel, good scientific and personal connections in this fascinating field of regenerative medicine, to which I'm very connected for many years now, not on a scientific level, but certainly in personal interest. And it is also our very good fortune to have with us Professor George Bailey, and it is my honor and pleasure to introduce him. Can you still hear me?
Yes.
Okay. So George Q. Daly is the Dean of Harvard Medical School and the Caroline Shields Walker Professor of Medicine, a physician scientist and a world-renowned authority on stem cell science and cancer biology. Daly earned his bachelor's degree, magna cum laude, from Harvard and his PhD in biology at MIT, working with the Nobelist David Baltimore. Daly has been professor of biological chemistry and molecular pharmacology at HMS, and was an investigator of Howard Hughes Medical Institute from 2008 until he resigned in 2017 upon assuming the deanship of Harvard Medical School. In addition to his groundbreaking research, Daly has been a principal figure in authoring international guidelines for stem cell research and its clinical translation, particularly through his work with the International Society for Stem Cell Research, where he served as a president. He has repeatedly testified before Congress and spoken in forums worldwide on the scientific and ethical dimensions of stem cell research and genome editing. Daly is a recipient of numerous awards and prizes, including an inaugural winner of the NIH Director's Pioneer Award for highly innovative research. He is an elected member of the National Academy of Medicine, the American Academy of Arts and Sciences, the Association of American Physicians, and the American Society for Clinical Investigation. So thank you very much, Dr. Daly, for being here with us today, and the floor is yours.
Thank you very much, Dr. Karmi, for that introduction. And it's an honor to be here in front of this new Israeli Academy of Medicine. And I'm pleased to see so many wonderful colleagues. I see Stefan. I'll say hello to Achia. And I think I saw Dr. Itzkovitz Eldor on earlier. And I very much am pleased by my many colleagues in Israel, and I look forward to a day when I can return again. And I am scheduled to come to the Israeli Stem Cell Society in September of 2022. So let's all hope that the world is back to normal by at least that time. I'm going to share my screen, and then I'll start my presentation. I'm going to be talking about work that has really been a central focus in my laboratory for more than 25 years. And that involves a vision that I shared very early with Dr. Itzkovitz Eldor, who shared the first isolates of human embryonic stem cells with my laboratory and set me on the path to trying to develop methodologies for deriving blood cells from pluripotent stem cells in a Petri dish. And that has evolved, and I'll be talking about some of our published work, but also the second half of my talk will be entirely devoted to unpublished work where we've been able to derive T cells and anticipate very rapid clinical translation. I do want to make some disclosures. I hold equity in a number of biotechnology companies, none of which are being represented here today, but I do hold patents on the iPS-derived blood products. I first have to say that I'm very pleased to see Achia Urbach, who was an absolutely terrific postdoc with me some years ago, who's now independent at Bar Ilan. Achia came with an interest in probing the application or the involvement of the Lin-28 RNA binding protein in germ cell tumorigenesis. And interestingly, and relevant to the presence of Dr. Benny Deckel, what we found instead were tumors of the kidneys. And together with Dr. Deckel, we were able to demonstrate indeed that the Lin-28 pathway is a major player in driving human Wilms tumor. And indeed, one of the companies that I mentioned at the top, 28/7 Therapeutics, is developing small molecule inhibitors of this pathway in hopes of being able to target tumors like Wilms and others. I owe a tremendous debt to Achia for his tremendous work in this area. So let me speak about pluripotent stem cell derived blood cells, and why would one be interested in trying to manufacture cells when we have a widely applied blood banking system where donors through altruistic acts come forward and donate their blood and blood products. We do understand that this system, though effective and economical, is fraught with its own challenges. Whenever we have a snowstorm here in Boston, our transplant program has to go on rationing of platelets in particular because donors aren't able to make it in. And so being able to manufacture blood from a petri dish would provide an inexhaustible and predictable supply of blood products. Furthermore, we know that there are pathogens that make their way into the blood supply, hepatitis C, HIV, more recently Zika. And being able to manufacture blood would provide for a pathogen-free resource. And then one could further define antigenically the types of cells, inexhaustible supply of O-negative red blood cells, for instance, or PLA1 positive or negative platelets. Also, there's a significant degree of variability in the blood supply. Donor to donor will have different amounts of red cells and platelets, and being able to manufacture really would give pharmaceutical grade consistency. And then of course, these would be offer the potential for off-the-shelf types of products rather than patient-specific. Now, I won't speak as much today about the work that we've done in making platelets, but platelets are actually in clinical translation and trials in Japan. We've recently made major advances in being able to produce red blood cells, in particular, the red blood cells that produce the mature adult beta globins, which have been a challenge, a barrier to pluripotent stem cell derivations in the past, and as well natural killer cells. I'll talk a little bit about our efforts to derive hematopoietic stem cells, and still we're tantalizingly close to realizing this potential, but still have work to do. And I'll speak quite a bit about our ability to generate T cells, which have multiple applications in the treatment of cancer through adoptive immunotherapy, or for T regulatory cells for graft versus host disease or autoimmunity. So one should start by saying, why is it so difficult? Laboratories around the globe are focused on directed differentiation of pluripotent stem cells into many, many different lineages. But the blood is particularly challenging, in part because blood development in the mammalian embryo occurs in waves. And here is shown in the mouse in the upper left, the first blood cells that arise in the mammalian embryo actually appear in the extraembryonic tissues of the yolk sac, the blood islands of the yolk sac. These we call primitive, and these are a particular type of cell, a predisposition to embryonic red blood cells, those cells that produce embryonic and fetal globins, which are oxygen avid and actually serve to pull the oxygen from the maternal blood system. There's also a series of yolk sac macrophages, which we now know actually are resident in tissues and perhaps in the brain throughout much of adult life. But the real blood cells that we'd like to define are that second wave. It's a definitive wave. These are the cells that become the source of all circulating blood throughout adult life. These are the cells that make lymphocytes, and they actually derive from a completely different mesodermal lineage that arises in the floor of the developing aorta from a specialized hemogenic endothelium, and I'll be talking more about that. Now, the default pathway of differentiation for pluripotent stem cells is into this primitive wave. And what we've had to learn, and this has been taught to us recently by Igor Slukvin and Gordon Keller's lab, is how to actually repattern the pluripotent stem cells so that they differentiate into this more definitive wave of hematopoiesis. So for many years, indeed, I, with Dr. Itzkovitz Eldor's help, actually secured the first NIH grant for studying human embryonic stem cells back in 2001. And at that time, we were able to coax the differentiation into blood products, blood cells, primarily CD34 positive, CD45 positive myeloid erythroid cells. And the first grant actually was predicated on testing the hypothesis that there was an intermediary hematopoietic stem cell, which was transient in culture. In fact, that hypothesis was wrong. The default pathway does not go through, in this primitive way, an HSC intermediate. We had to learn instead how to pattern this hemogenic intermediate, this intraembryonic aortic source of the definitive hematopoietic stem cells. And what was taught to us by the painstaking work of Gordon Keller and Igor Slukvin is that here on the lower right, if you want to direct the differentiation of pluripotent stem cells, one has to replicate the polarity induction that typically accompanies gastrulation in the primitive streak. And they showed us that you could do this by activating the Wnt beta-catenin signaling pathway and inhibiting elements of activin and nodal so that you would skew the differentiated population towards more so-called posterior blood fates, and that included the hemogenic endothelium. Now, unfortunately, the Keller lab and our lab has attempted to directly engraft lethally irradiated mice with these hemogenic endothelial cells, the immediate precursors of hematopoietic stem cells, but we've never been able to achieve engraftment, suggesting that there's a significant distance, if you will, epigenetic distance, between what we can make of these cells and their commitment to hematopoietic potential. And this led us, and this was work directed by a postdoc, Ryo Sugimura, to attempt a screening strategy to identify transcription factors, which when ectopically expressed in hemogenic endothelium, would drive them towards hematopoietic stem cell fate. This is published, so I'll simply give the data, but we took a Yamanaka-like approach to identify a library of some 20 transcription factors. And we were able to, as seen on the upper left, introduce this library into hemogenic endothelial precursors and select in engrafted mice 4 blood cells that actually engrafted not only the primary mice, but could be transferred and engrafted into secondary mice. And by flow cytometry analysis of the bone marrow of those secondary mice, we saw elements of human hematopoietic stem cells, human red blood cells, human white blood cells, human B cells, and human T cells. Furthermore, we could explore those lymphocytes, and the lymphocytes being the definitive wave of cells, those cells that we know have this long-lived adult definitive potential. And the mice were in fact engrafted in their thymus with human CD4, CD8 double positive T cells in general. There seemed to be a block in conversion to single positive CD4, CD8 cells. But these were indeed alpha beta T cell receptor expressing T cells. They had a very broad repertoire of the T cell receptor as shown by immunosequencing. And in a very definitive experiment, we could actually demonstrate that there was not only human immunoglobulin in the serum of these mice, but we could see a boostable immune response to an ovalbumin challenge of both an IgM and an IgG cell subtype, suggesting that there was immune function as well as T and B cell cooperation. Now, further looking towards the molecular signatures of these human hematopoietic stem cell-like cells, we performed single-cell RNA sequencing, compared them to cord blood hematopoietic stem cells. And I think it's important to point out that by t-SNE analysis, here in blue, the iPS-derived hematopoietic stem cells are indeed distinct from cord blood-derived hematopoietic stem cells. There are rare elements of iPS derivatives that do seem to overlap. And further, if we look at the upper right, if we supervise that analysis and look specifically at 62 hematopoietic transcription factors that represent a signature of hematopoietic stem cells, indeed that core program is indeed overlapping between the iPS derivatives and the cord blood derivatives. So I think it's fair to say that we have the function and some of the molecular identity of hematopoietic stem cells, but we're continuing to evolve this system to try to achieve an ever closer approximation of natural hematopoietic stem cell function. That work was published some years ago now. An interesting question was then taken to the next stage by a terrific graduate student, Linda Vo. She asked what was the barrier between these primitive cells and their definitive HSC counterparts. And she asked a question, whether or not she could identify through a genetic screen factors that repressed the multipotency, the lymphoid myeloid multipotency of definitive hematopoietic stem cells. And she configured a screen using a library that we had previously used to great effect to probe the barriers to somatic cell reprogramming, a series of chromatin modifying enzymes listed here. This library is available from the AdGene resource. She then did the following experiment of introducing this library into these hematopoietic progenitors, these primitive progenitors that were restricted in multipotency. And then she measured for the ability of these knockdown modifier targets to produce either T cells or B cells. And the experiments were really quite revealing. The top hit in this genetic screen was the knockdown of the histone methyltransferase EZH1. And here you can see by flow cytometry a significant augmentation of the development of CD4, CD8 double positive T cells. And in looking at the overlap of T and B cell fate, the one factor that seemed to be strongest in stimulating both lymphoid derivatives was EZH1 itself. So EZH1, then we subjected these cells to RNA sequencing, and you can see here a quite distinctive array of genes, which by Go analysis are highly linked to a lymphoid signature. So the knockdown of EZH1 leads to the upregulation of lymphoid genes. Furthermore, by ATAC sequencing, a strategy that asks for the evaluation of open chromatin, presumably transcriptionally active chromatin, once again, EZH1 knockdown led to the opening of gene sets that are involved in lymphocyte and T cell activation. So these data showed that this histone methyltransferase, which is part of a polycomb repressive complex that is broadly repressive of chromatin, that this was sitting on a diversified array of lymphocyte targets and silencing them, and that this helped to explain the inhibition of lymphoid potential in those primitive and mid-gestation embryonic populations of blood. This was also published some years ago. Now, this work is really what has led us to be able to now move to the next stage, which is to exploit this ability to activate lymphocyte potential to develop iPS or ES-derived T cells for adoptive immunotherapy of cancer. This has really been a revolution in the making. Being able to take a patient's own T cells is shown here. Here's an individual, let's say, with a lymphoid malignancy like acute lymphocytic leukemia, one harvests the T cells and then can engineer them with a chimeric antigen receptor, which targets these T cells to kill the lymphoid malignancy. One expands them and then treats the patient with chemotherapy and reinfuses these T cells. This CAR T cell therapy has turned what was previously a highly refractory and highly fatal malignancy into one that induces complete remission in a very significant percentage of patients. The problem is that because this is individualized, patient-specific, very, very labor-intensive, these are among the most expensive therapies in all of medicine. And we have felt, as have others, that if we could derive T cells off the shelf from a universal donor source like embryonic or induced pluripotent stem cells, we could democratize the treatment, make it more widely available, and ideally bring down the cost. This experiment was initially done by Michel Sadelain, one of the major figures in driving CAR T cell therapy. This was a proof of principle experiment published in 2013, starting from iPS cells that were derived from patients' T lymphocytes. So these iPS cells would carry a already rearranged T cell receptor, but in order to bypass that specificity and introduce CD19 specificity, and CAR antibody, or I'm sorry, a chimeric antigen receptor was engineered into these iPS cells, and then they were differentiated into T cells using a standard platform of a notch ligand expressing stroma. This showed activity. Interestingly, the iPS-derived T cells were somewhat less effective than peripheral blood-derived T cells. Now, in this experiment, in a humanized mouse model of CD19 positive lymphoma, the peripheral blood-derived T cells, which might be otherwise used for actual therapy, protected 100% of the mice, whereas the parental lymphoma kills these mice quite rapidly. The good news was that the iPS-derived CAR T cells did protect most of the mice, but that protection was less robust than the peripheral blood. And in analyzing the nature of these cells, it was clear that the iPS-derived T cells had the signature of gamma delta rather than alpha beta positive T cells. And the gamma delta cells tend to resemble a more innate cell type, and they tend to be less robust in tumor cell killing. So the goal that we have set out is to use our EZH1 knockdown strategy to derive more adult-like and hopefully more robust T cells for this therapy. And this is a work of a postdoc currently in the lab who has really transformed our ability to use EZH1 knockdown, as here shown in the upper schema, through a stromal-free in vitro differentiation platform to derive single positive, what we are calling easy T cells. We can see after several weeks of differentiation, a strong signal of CD5, CD7 double positive T cells, and the production of primarily single positive CD8 T cells with a modest contribution of CD4 positive T cells. Now, there is some evidence that the most effective formulations of peripheral blood-derived CAR T cells actually harbor a larger percentage of CD4 positive cells. And currently we're involved in experiments testing whether we can stimulate the CD4 population here. But even with the population as it is, we can demonstrate that our EZ T cells are expressing the alpha beta T cell receptor, They are very much depleted in the gamma delta T cell receptor, which was characteristic of the original iPS-derived T cells that the Catalan group developed. And furthermore, they do not express the markers of immaturity that are expressed on gamma delta T cells, and they express a classical CD8 alpha beta dimer, which is different from the Catalan T cells, which, as you can see here on the lower right, were predisposed to express a CD8 alpha homodimer. By RNA sequencing, looking at T cell signature genes, once again, the easy T cells are most closely aligned with a classic alpha beta T cell, quite distinct from the gamma delta. And this is also seen if one supervises the analysis based on those genes expressed in alpha beta T cells. So by surface antigens, as measured by flow cytometry, and by RNA sequencing, these look like very similar to adult peripheral blood derived T cells. So then working with Marcella Moss from the Mass General, her laboratory is expert at CAR T cell therapeutics. We were able to engineer a second generation CD19 directed chimeric antigen receptor into our T cell platform. The expression of this CAR was followed by the co-expression of an M-Cherry fluorescent marker. We could show that indeed these CAR-loaded T cells expand. We were hopeful that the CAR might actually stimulate greater expansion than the untransduced controls, but in fact, they're roughly the same. We can show that the CAR-loaded T cells undergo T cell activation by expression of the CD69 activation marker in response to treatments with ionophores and 4-vol esters. And then finally, in an in vitro degranulation assay, we can show that when these CAR-loaded T cells are challenged with CD19 expressing leukemia targets, we see a marked degranulation showing activity of these T cells against the leukemic targets. Then working with Marcella's lab, we were able to perform a direct head-to-head comparison with standard peripheral blood-derived CAR T cells against our easy T cells derived from iPS. And in a dose titration, we could see equal effector cell killing against both the CD19 NOM6 and JCO1 tumor cells. And then finally, in a demonstration that's very preliminary, but we were able to do a tumor cell kill, essentially a tumor cell titration in a mouse model of CD19 positive lymphoid malignancy. I think you can see here as we escalate the dose, the incidence of this lymphoma in these mice occurs at earlier and earlier stages. And we chose a dose of 1 million tumor cells and then performed an experiment where we induced the tumor in the mice and allowed the tumor to form. And then at day 21, after quite a significant tumor burden, we injected the iPS-derived CAR T cells. And whereas you can see the untreated mice developed a significant tumor burden by day 28, going on to succumb to this malignancy by day 35, the CAR-treated animals show the establishment of disease at day 21 when the injection occurs, and then the resolution of disease, and indeed these mice remain disease-free, showing that the CAR is able to eradicate. Now, this is just a pilot experiment. A larger number of mice are currently being tested, but we're further encouraged by the fact that at day 49, we can identify in these mice the CD3 positive, M cherry positive CAR T cells, knowing that these iPS-derived derivatives are in fact persisting in these mice and correlate with the extinction of disease. So let me just finish by saying that I've shown you that pluripotent stem cells tend to default into the primitive pathway for blood formation. And in the experiments aimed at developing T cells for adoptive T cell therapy, the classical default pathway generates immature T cells. We launched genetic screens to identify chromatin modifying factors and identified EZH1 as a factor that limits lymphoid fate. And now we also have independent chemical screens which identify small molecules that can also promote definitive lymphoid potential. We've been able to use genetic knockdown of EZH1 and a stromal-free T cell differentiation system to develop CD4 and CD8 positive T cells. I did not show the data today, but in fact, just in my group meeting before this seminar, my postdoc demonstrated that he can also divert these EZH1 knockdown cells very, very effectively into pure populations of CD56 positive NK effector cells. By molecular analysis, we actually are quite confident that these EZ T cells are very comparable to peripheral blood-derived T cells. And we've been able to demonstrate that we can effectively load them with anti-CD19 CARs, and we can demonstrate proof of principle for tumor cell killing, both in vitro and in vivo. And currently, we are expanding that mouse model of pluripotent stem cell derived T cells, and we are performing head-to-head comparisons on a per cell basis for the EZ T cells compared to traditional iPS-derived gamma delta T cells, as well as testing NK cells. And we are anticipating the prospects for manufacturing scale-up for potential clinical applications. Now, what I have not spoken to, and could be a talk in and of itself, is the fact that these T cells would be subject to, if we take them off the shelf and try to treat any random patient, would indeed be subject to both rejection by the host immune system, which could be dampened down by pretreatment conditioning, but also we would worry that the existing T cell receptor repertoire could set up a situation of the CAR T inducing a graft versus host disease in the recipient. And there are a number of groups, David Russell, Chad Cowan, and others, who have worked on strategies for immune cell cloaking. That is to take the pluripotent stem cells, knock out using CRISPR technology, the various immune recognition molecules, such that these cells would then be, in a sense, invisible to the host immune system. That's still a technology in need of further development, but the combination of cloaking and directed differentiation of blood cells from a Petri dish, I think, gives us the prospect of off-the-shelf blood and immune therapies. So let me finish so we have time for questions. I want to acknowledge the collaboration with the Marcella Moss Lab on the CAR T cell platform, and point out that since becoming dean, I have had to shrink my lab considerably and have now partnered with two close colleagues here listed at our annual retreat, Trista North and Torsten Schlager, so that there still remains quite a robust community to collaborate and to bring these platforms forward. So I'm gonna stop sharing and thank you for your attention and take any questions.
So we have one question, first of all, on the chat. This is amazing. How sure are you with EZH1 cells in terms of tumorigenic development? Can you combine universal donors with EZH1 manipulation?
So this question from Dr. Deckel may take note of the fact that EZH2 knockout or EZH2 mutation is associated with lymphoid malignancy. I have not seen evidence that EZH1 deficiency is likewise associated with malignancy. We have examined EZH1 knockout cells. They do have some defects in terminal blood cell development, but what we have adapted to is now a transient EZH1 knockdown. So we're hopeful that this intervention just transiently is enough to reactivate T cell potential without promoting T cell malignancy. We've only done pilot experiments to date showing that we can in fact knock out the endogenous T cell receptor and still load these cells with a chimeric antigen receptor and derive T cell function. What we have not yet fully explored is whether we can engineer HLA mimics into these cells so that we could avoid, let's say, an NK cell response. We presume, and we're in fact in conversations with groups that work on this cloaking strategy so that we could actually merge these two technologies to imagine an off-the-shelf product.
Thank you. It could be that the questions are just a little too complicated to write them on the chat. So I'm going to actually allow, if anybody has a question that they want to ask orally, you can just unmute yourself and ask Professor Daly. Is there anyone that wishes to do so?
I might add the commentary that I don't know how many of you are, I mean, I know Dr. Deckel is going to speak about kidney development, and there's a very fertile area of being able to direct the differentiation of pluripotent cells into kidneys and the formation of kidney organoids and the like. I think many, many different tissues see the same phenomena, which is that the default pathway of early differentiation of pluripotent stem cells is towards these embryonic lineages. And that in cases, let's say for neural development, parts of the brain, whether it's dopaminergic neurons or cholinergic neurons of the forebrain, the fact is that the brain really is an embryonic organ. I mean, those cells were largely laid down during fetal development. And so the scientists and communities that focused first on nerve cell development really had an advantage because they were already working with this primitive program. We've had a challenge because we've got to pattern definitive adult blood cells, and those have really undergone quite a significant developmental maturation. And we have to mimic that in the Petri dish. And so I do suspect that this has been something that's confronted those who work on beta cell development. There's some evidence that the Lin28 protein itself has to be downregulated in order to convert what would otherwise be embryonic or fetal beta cells into adult equivalent beta cells. That's work from Robert Blaylock's lab. And I might imagine it would also impact kidney lineages. As we've shown that the Lin28 is involved in the cessation of development so that you can move from expansion of kidney progenitors into sort of the nephrogenic phase. And so I think this is a theme that you'll witness again and again.
Thank you. We have another question from Ari Gargir. Can you please elaborate on your recent understanding and progress in red blood cell culturing?
Yes, if any of you are reviewers on our paper, we ask for your indulgence. We're going to come back soon with improvements after a set of challenging reviews. There are a number of elements that have allowed us to take pluripotent stem cells and developmentally mature them through a stage of globin switching. The classical problem with red cell development has always been that you get this massive expansion of cells that express embryonic and gamma globin. And I can't say that we had any really brilliant insights molecularly. We tried to manipulate the BCL11A repressor of fetal globin. We tried to manipulate a number of other genetic pathways, but what we ended up doing was a sort of brute force strategy looking at different culture conditions, and in particular, prolonged culture in the presence of human plasma and human serum. And that seemed to trigger a significant degree of both beta globin switching as well as enucleation. We've actually been able to use that platform to model human sickle cell anemia. One of the problems with getting cells to sickle is if they express a lot of this fetal globin, they won't sickle. We now have evidence in our cultures that at least some subpopulation of these red cells actually undergo sickling. So we presume that they have downregulated their gamma, and by molecular and HPLC analysis, we know they're upregulating the sickle form. So we're pretty enthusiastic that we can move that forward. Of course, then the challenge becomes, can you manufacture at scale? Because otherwise the cost of goods gets so that, you know, you could make a unit of red blood cells, but it might cost you $15,000 US, which isn't competitive with donor. So there's still some significant manufacturing challenges.
I think we have time for one or two more questions. So can you see, Professor Daly, the question from Professor Shai Israeli? I'm just afraid I'm going to angle the word after definitive. So
yeah, I have to say, so this is a very thoughtful observation that there are distinctions between the fetal liver hematopoietic stem cells and adult bone marrow derived hematopoietic stem cells. Typically, if one does a competitive head-to-head repopulation, the fetal cells will win out. They are more developmentally robust. We have not gotten to the point where we can make a direct comparison. We haven't done that experiment, but I'm going to presume that we're probably generating something that's more akin to a fetal hematopoietic stem cell than an adult hematopoietic stem cell, but that's really based on just my intuition more than actual data. Oh my gosh, Mitch Swapper, how are you? Good to see you.
Great. Do we have any more questions?
Co-trainees.
Hi, George. Great talk.
Hey, how are you? Good to see you.
You too. You too.
Great.
Fantastic.
Okay, then Professor Carmi, I'm going to give the mic back to you. We can hear you.
You sure?
Yes.
Okay. Oh, so many challenges. So thank you so much, Dr. Daly, for, even to me, it was a fascinating presentation. Thank you very much. And before I introduce to you our next speaker, I would very much like to address and announce the presence of Professor George Griffin. He is the president of the Federation of European Medical Academies. Hi, George. Good to see you here. And now for our own Professor Benjamin Deckel. Benny Deckel, MD, PhD, is the director of the Pediatric Stem Cell Research Institute and the chief of pediatric nephrology at the Edmond and Lily Safra Children's Hospital, Sheba Medical Center. He's a professor of pediatrics, human genetics, and biochemistry, and the incumbent of the Pearl and Dr. Yechezkel Kleiman, Chair of Urology at the Sackler School of Medicine, Tel Aviv University. Dekel received his MD degree from the Technion and a PhD from Weizmann Institute, all with distinction. He completed the pediatric residency at Sheba, postdoctoral fellowship in stem cell biology at the Weizmann Institute, and a pediatric nephrology fellowship at the Schneider Medical Center. Dekel served also as a visiting professor at the Institute of Stem Cell Biology and Regenerative Medicine, Stanford University. Benny is known internationally as one of the most innovative and recognized investigators in the field of human renal stem cell biology, cell therapy, and renal regenerative medicine. Professor Dekel is an elected member of the American Society of Clinical Investigation, the American Society of Pediatric Research, and was recently elected as the inaugural member of our Israel Academy of Sciences in Medicine. Professor Dekel has received multiple awards, among which are the Yehudim Prize for Excellence in Cancer Research and the Israel Medical Association Prize for Outstanding Medical Scientific Contributions. Benny, you have the floor.
Okay, thank you for the kind words. And of course, speaking after George is quite difficult, but I'll try anyhow. I'll share screen. Just a minute. Okay, so here we are. We're going from blood to kidneys. You know, blood and kidneys develop, the definitive site of hematopoiesis in the zebrafish is the kidney, by the way. So that's a link. And I'm going to take you to end-stage renal disease, what we see in the clinic. And of course, it's the inabilities of the kidneys to remove waste products and excess fluid from the blood. And you have to really note that when you develop chronic kidney disease, it's really can be classified in stages, right? Where you draw the creatinine clearance. This is the filtration product of the kidney, and you define the level of chronic kidney disease by blood and urine measurements. And you can see how chronic kidney disease is really worldwide. It's an epidemic, of course, with hypertension and diabetic nephropathy and so forth. We usually see patients in the pediatric ward with, of course, renal malformations that develop chronic kidney disease and turn into end-stage renal disease. Now, importantly, this is a critical phase, CKD4 to CKD5. Now, here you're in the office, right? You have 15 to 25% of your renal function reserved, and you still can be getting erythropoietin, you can be uplifting your calcium levels, reducing your phosphor levels, you can take care of your metabolic acidosis. But here, stage 5, this is really what we say terminal renal disease, and you need something else. You need renal replacement therapy. And of course, you all know dialysis, which is a well-known paradigm in medicine. Transplantation, Joe Morey did it about 60 years ago, right, with the first transplant, and huge waiting list. Well, I think there was about 100,000 people waiting for a renal allograft in the U.S., and it's, you know, just becoming increasingly demanding, the need for renal allografts. So we needed some kind of paradigm shift, and we call this the kidney repair shop, or the kidney garage, where we need to grow, replace, or rejuvenate parts of the kidneys, you know, nephron cells, nephron parts, whole nephrons. And this will be mainly the topic of my talk. And this is regenerative medicine. Regenerative medicine typically uses combinations of cells, biomaterials, and biomolecules to rejuvenate or replace damaged or diseased tissue. And this is a very nice scheme of the different participants in Regen Med, right? The cells, the biomolecules, the biomaterials. We are mainly focused on the cells, okay? So I'm going to discuss cells, and this is, I would say, the start of regenerative medicine. You pull you add to the cells a biomolecule, a biomaterial, but the cells are the essence of cellular therapeutics and tissue engineering. So you're going to hear about cells. And what would be the optimal kidney forming cell? This is an important slide. One, it should be expandable, as Dr. Daly showed. It should be long-lived, self-renewing over many generations in the Petri dish. So one thing would be expandable. Second, it should maintain renal potential. Now, it should be preferably multipotent, as much as lineages as possible in the kidney. You know, maybe all nephron cell types. Transplantable, we want, these are not hematopoietic cells, right? They're not into the blood. They need to engraft in a tissue environment. So they need to engraft efficiently. They need to generate healthy tissues and disease models. And of course, they need to be safe, non-tumorigenic, and that's an important quality. So what would be the starting material for a kidney-forming stem progenitor cells? So, of course, we have the pluripotent stem cells, and I draw the line here. These guys would be pluripotent stem cells, and here, this is a transdifferentiation protocol. Now, why am I drawing the line here? Because here you would need to differentiate the pluripotent stem cells towards a renal lineage by a differentiation protocol. And here you would need to transdifferentiate, take another mature cells, put in transcription factors of the renal lineage, and sort of take them across lineages and make kidney cells. So here, in contrast, the human embryonic kidney or the adult kidney, here you need to find the cells. You don't need to differentiate them. You need to find them and keep them in culture and cultivate them and expand them. And I think the basis of understanding kidney-forming stem cells relies in the human embryonic kidney. And so I'll just show you published data where we actually started our endeavor with the human embryonic kidney. And this is the site of nephron production. You know, the metanephric kidney appears at around the 5th week of human gestation. And it's composed of two main components, the metanephric mesenchymal cells, which undergo what we call a mesenchymal to epithelial conversion. These guys know how to become the epithelial cells of the nephron, the entire nephron. The ureteric bud epithelia are a branch of the Wolffian duct. They invade the mesenchymal cells, the metanephric mesenchymal cells, and start branching. So this is a different process. It's called branching morphogenesis, and the ureteric bud epithelia gives rise to the collecting system of the kidney. Some of the ureter and also maybe some of the bladder, the trigon of the bladder. So these are two different lineages that you would need to reconstruct or you need to manipulate in vitro. So I'm going to take you to developmental biology of the kidney. This is a section of the human metanephric kidney, very early human fetal kidney. Here you see these are actually stem cells of the kidney. They will form nephrons. This is the ureteric bud. They talk to each other, and you see a nephron anlage. This is the beginning of nephron differentiation. You see epithelial cells, you see polarization, and these will become the adult nephrons. And thanks to our friends, the developmental biologists, some great contributions in understanding nephrogenesis, or the differentiation of the embryonic renal stem progenitor cell pool. These are the metanephric cells. This is the ureteric bud, again, the branch of the Wolffian duct, a Wnt9b signal, and then these cells start to condensate on the ureteric bud. You can see what we call the kept mesenchyme cells. They express a bunch of renal transcription factors, 6-2, OSO1, PAX2, cell1, Lin28 is present here. They secrete GDNF, they express vimentin, they're mesenchymal cells, and then they start aggregating, as I showed you in the picture before. We generate a pretubular aggregate, they secrete a Wnt4. And for instance, if you have a Wnt4 mutation, which we described quite a while ago, you have a renal malformation, right? Because the nephrogenesis stops. This is a comma-shaped body, what we call the nephron anlage, and this is the mature nephron right here with all cell types. Now, this process takes around 34 to 35 weeks of human gestations. we generate nephrons only in fetal life, and this is an important message. We don't have neonephrogenesis in adult life, just in fetal life. And these guys, these renal transcription factors actually go away. Well, you see WT1 in the podocyte, you can see PAX2 in some tubular cells, but mainly they go away. And a method to bring up or to reprogram adult cells would be to really reintroduce these embryonic renal lineage transcription factors in the adult kidney. So this is nephrogenesis. And what we did, and this is looking at other organs, just like the hematopoietic system, where you have a nice hierarchy of cells, this is a tour de force of prospective isolation of human renal developmental progenitors of the human fetal kidney. And here, it's just like the hematopoietic system. You sort out a specific lineage or a specific developmental progenitor with just three surface markers. This would be NCAM and CD133 and EpCAM. So I want you to, NCAM is CD56. It's also expressed on NK cells, but NCAM high cells, CD133 cells, are early progenitors. And then EpCAM starts, EpCAM dim, just like the hematopoietic system. And then there's EpCAM bright. And this is not a course in renal biology. It's just, I want to illustrate how you can really isolate a specific developmental progenitor in a specific time point with just three surface markers. And this has been published. Now you can really mimic the mesenchymal to epithelial transition in the cartridges, and you can really pinpoint these developmental stages with the three transcription factors. And with the three surface markers, I'm sorry. Transcription factors, of course, are irrelevant for sorting, and of course, you would need the surface markers. So this is what we did in the lab. We got human fetal kidneys on a regular basis. We collaborate with the OBGYN around different hospitals in Israel, and we can get a collection of human fetal kidneys. These are aborted fetuses. We can also use Wilms tumor, which is sort of recapitulates the developmental lineages. And we discovered CD56, we discovered NCAM1. So we go to the machine, to the cell sorter, and we purify an NCAM1 population as opposed to an NCAM minus population, what we call a stem progenitor population compared to a non-stem. We culture them, we run gene expression, and we asked ourselves, do they express the renal transcription factors that we know that exist in renal development? And we do the culture assays, we do the clonogenic assay to show self-renewal over generations. And these are in vivo assays, which are crucial. We need to generate a mouse model of disease and inject cells into these mice and show regeneration in case of isolation of NCAM1 progenitors from the human fetal kidneys. Now, if you isolate these NCAM1 progenitors from Wilms tumors, of course, you get tumor initiation, you get tumor growth. And by the way, there was a clinical trial with an anti-NCAM1 ADC developed at Immunogen in Cambridge, which they tried to really combat Wilms tumor relapse with an anti-NCAM1 anti-drug conjugate. So this is just an illustration of how bedside research can reach the clinic. So this is actually the first example of human cells in mice that can halt the progression. These are human fetal kidney progenitor cells transplanted into a mouse model that shows progressive renal disease. You can see it here, chronic kidney disease. You put the cells into the mouse, and you can really influence the progression of the disease, of the chronic kidney disease progression. I'll elaborate about this a bit later. But as we said, why do we need to take aborted human fetal kidneys? I mean, it's ethically disconcerting. It's really hard to get. You need to, the patients need to sign so we can change pluripotent stem cells into kidney organoids. And this is work we're doing in collaboration with Benjamin Friedman and WashU and also Achia, Orbach at Bar Ilan, where we can take the early stages of pluripotent stem cells and have this differentiation protocol towards a kidney organoid, which is what I like to say a fetal-like kidney tissue. Okay, it's not a mature kidney tissue. This is the organ. This is the human fetal kidney, and this is the human mini kidney organoid. So you run a transcription analysis, you run a transcriptome, a bulk transcriptome on this organoid, and it mimics around, I would say, 10-week human fetal kidney. So the lineages are not fully mature there, and you have to appreciate that. I mean, pluripotent stem cells are extremely cool, and you know, these are iPS cells, these are autologous, of course, but still the organoid that develops contains developmental lineage, and I want to emphasize that. But this is, again, this is unpublished work with Ben Friedman and Achia Orbach, where we actually analyzed the biomarker system, the ENCOM, CD3, EPCOM system we discovered in the human fetal kidney, and we show it to be sustained in iPS kidney organoids. So here is ENCOM high, CD133. You can see CD133 is really correlates with LTL. This is a differentiation marker of the proximal tube. So again, an ENCOM plus CD133 minus sort would give you the stem cells. And I was very glad to see that, because sort of it tells you that you might go to the what we call the iPS fetal-like kidney tissue, and then differentiate from the fetal-like kidney tissue your early nephron stem cells and culture them in the Petri dish. So these surface markers that we discovered back in 2013 are very relevant to the iPS kidney organoids, and you can use them to derive progenitor population from kidney organoids. Another way to sort of get rid of the human fetal kidney, which upset quite a lot, is to use amniocentesis and use of fetal urine. And remember, amniocentesis actually is mostly urine at around 18 weeks of gestation. So we can do amniocentesis at around 20 weeks with the, again, we can collect many amniocentesis and amniotic fluids at Sheba and other OBGYN centers. And once you have amnion, you can culture them. And this is new data. Of course, this is Oren Planciano's. He just, a postdoc of mine, he opened a lab here at Sheba, and he's using the amnion as a source for human fetal kidney-like organoids, again, derived from fetal urine. So we might not really need to derive developmental lineage. We can go to iPS cells, generate the kidney organoid, sort out the progenitors according to our markers, or just go to the fetal urine, derive organoids from the fetal urine, which is can be derived from the amnion, from the amniotic fluid, and culture the cells as organoids, and again, take out the progenitors for regeneration. So this was, the first part was really to discuss the way, you know, the human fetal kidney, the iPS derived kidney tissues, and the maybe the amnion kidney organoids for generating developmental lineages, renal lineage for regeneration. But I think the main challenge is with the adult kidney. And I think the adult kidney could go first to the clinic, because these would be mature cells, and these would be your own cells without any, you know, just your own cells without any differentiation protocols, without, and you can take them in a non-invasive manner. I'll show you in a minute. So the adult kidney is a major challenge in my opinion. But remember, no nephrons are formed in adult life. We get a 4-centimeter kidney, and we have a 12-centimeter kidney in the adulthood, but it's mostly, you know, just elongation of the nephrons. The fish has these embryonic progenitors left in adult life, so physiologically, the fish can generate nephrons along in adult life, but the mammals cannot, and this is nephron loss and lack of nephron generation. And the fish has these nephron progenitors again during pathology, generates new nephrons, and has you know, it has a bunch of nephrons working for him. So we're not fish, we're mammals. And this is the kidney, of course. And remember, we generate urine in the kidney. You have this cartoon where urine is just being is flown through the kidney, and this is the urine. We urinate around 70,000 cells, kidney cells per hour. So think about it. If we didn't have a mechanism for cell turnover or cell replacement in the adult kidney, I mean, we don't grow new nephrons, but we need some kind of mechanism to generate new cells. Otherwise, you would urinate your kidneys out, practically. So this is what we discovered. This started with my sabbatical at Stanford, and I was lucky to get the rainbow mice. Rainbow mice are multicolored mice where you can really generate a gene color in a specific cell and follow clonal evolution. You can, by injection tamoxifen, you can set a specific red or a specific blue or a specific green and follow clonal evolution in any organ you might want to use. And this is the kidney. So what we showed is this. You have clones grown in each segment of the kidney. It's complicated. This is a local regeneration method. You don't see it. I mean, in fetal life, you need to generate nephrons. In adult life, you need to maintain them. And what we discovered is that you have this local regenerative mechanism in each segment of the kidney. So the proximal tube grows clones in the proximal tubes, and the loop of Henle grows clones in the loop of Henle, and the distal tube and the collecting system and so forth. And this compensates for the loss of 70,000 epithelial cells per hour. These are mature renal epithelial cells that behave as stem cells. They receive a Wnt signal from a neighboring cell and start clonally proliferating. So we call them Wnt responsive clones, but they're not professional stem cells. They weren't born as professional stem cells. They're facultative stem cells. They're mature renal cells that receive a signal and start clonally dividing, generating clones of cells. This is how it looks. This is how it looked in the rainbow. You see, this is a clone, this is a blue clone, and this is a red clone. But it was always segment specific. There's no multipotent stem cells in the adult kidney. There are unipotential progenitors in each segment that operate locally, like local guard for local regeneration and preservation of kidney loss. So we had to discover first the mechanism of how the adult kidney grows. So what do you do with this information? I mean, what do you do? How do you take this, this information of these clonal progenitors in each segment of the kidney, of the adult kidney, to the relevance, make it relevant to all patients? So what we did is this. We started off with the adult kidney and we extracted adult kidney cells. Now what we do is we expand the cells first in 2D cultures. We call these human kidney epithelial cells. And they explore, you can expand them. They differentiate, they undergo EMT, but then we generate spheroids. Actually, we generated spheroids. It was Ella Boujor's PhD study, and we started seeing these spheroid cultures in vitro, and this is a spheroid which is actually a ball of kidney cells floating in your petri dish. Remember that once the spheroid develops, cell cycle goes down. So you need first to expand the number of spheroids by doing the 2D expansion protocol. But the spheroid has the progenitors for each and every segment of the adult kidney. The spheroid has the progenitor for the proximal tube. It has the progenitor for the distal tube, the collecting system, and actually you can use it to generate different parts of the kidney. As I told you, there's not a single multipotent stem cell in the adult kidney. You would need to use a bunch of multiple heterogeneous lineage progenitors to do the work. And this is this is how it looks. This is just staining of the spheroids. I can show you, it doesn't mean a lot to you, but these are markers of different segments of the adult kidney, which are representative in these ex vivo adult kidney spheroids regrow in the petri dish. Another interesting observation is when we generate the nephrosphere, called the nephrosphere, you actually see renal developmental programs being upregulated in the petri dish. I mean, you do a 2D expansion and then you generate the kidney spheroids with a specific media, specific protocol. You start seeing how the kidney is rebuilt in the petri dish with activation of these kidney development molecules. Kidney epithelial genes are going up. Just you mimic renal development in the petri dish. So we have these ex vivo expanded kidney spheroids. We need to do better. And this is just, this is now in revision. We want to even get more cells. We need billions of cells for regeneration. We need to upscale. So when we do the 2D expansion with the adult kidney, the human kidney epithelial cells, we put in an OCT4, which is a pluripotent stem cell gene. And then we can really, on the coronal level, we can get much more, we can get upscaling of even more cells to like, P15, higher generations of culturing. And then we generate the spheroid. And again, the spheroid has you see different colors because it has the progenitors for each segment. And this is how it looks in the petri dish. And when you transplant these, you can get different segments. You get a proximal tube, you get a loop of Henle, you get a distal tube in the mouse. And this is the mouse model we're using. We used it when we transplanted human fetal kidney progenitors. This is, we work on this model because it's a stringent kidney model. This is the 5/6 nephrectomy. We take 5/6 of the kidney. Then you wait a week, you do the contralateral nephrectomy, and you're left with the remnant. We call it the remnant kidney model, which mimics a progressive renal disease. And then we transplant. These are human kidney spheroids into the remnant kidney model. And look what happens when you inject vascularly. You put these cells, I mean, if you put them IV, they would end up in the lungs. So don't try to put, if you want to hit the kidneys, don't try to put cells IV because they would be trapped in the lungs. But we wanted to do something, different. We did transplantations through the renal artery, which we said would be, a good way to bring about cells into the kidney. So you can see them actually stuck in the glomeruli. You can see that when you infuse the cells into the vascular compartment, they don't cross. And this wasn't our meaning. This was our meaning. This was actually what we call direct injection. You infiltrate the cortex and you transplant the kidney spheroids, and actually they know how to self-organize into a mash of renal tubules. So this is you can see these are all human cells on the human tubules on the background of the mouse. This is staining, for instance, for CD3. This is staining for EMA. This is a distal marker. You see a bunch of distal markers that are prominent in the in these kidney tubules. So you can generate both proximal and distal human tubules in the mouse. And what is neat is that we were really lucky to get CKD kidneys. I mean, sometimes the urologists operate and take out end-stage kidney disease, and they send it over to you because they have polycystic kidney disease. They're infected, or they have stones, so they send you discarded material, and you could actually really retrieve the human kidney spheroids from these end-stage kidneys, generate the spheroids, expand them, and put them into mice. So you can do it both from healthy and end-stage kidney disease, and this has been published. Again, you can see de novo structure formation, okay? This is just a tubular network that is generated in the mouse, the human tubular network generated in the mouse. And here you see specific cell replacement. You see a mouse tubule with a human cell, or a part of a human tubule, a part of a mouse tubule with human cells. So this is both de novo structure formation and cell replacement. And this is transplantation, again, into the 5/6 nephrectomy mouse model. Higher magnification, again, tubes and You see, this is cell replacement. This is de novo structure formation. This confocal 3D Z stacking, I want to just try to convey that there might be tubular connections. This is HLA staining, so you might be getting tubular connection, and actually these engrafted tubules can really connect with the host tubules, but we're not, you know, this needs to be confirmed. It's really hard to show these connections in a definite way, but this at least gives you some hints of connections between HLA plus tubes and HLA minus tubes, mouse tubes. So this is the immunostaining we did after kidney spheroid transplantation. But this is the protocol of extraction of kidney cells, 2D expression, 3D kidney spheroids, in vivo functional assays, and the therapeutic effects that I'll show you in a minute that appear. But the mechanism of the action of these cells, it's not just about generation of renal parenchyma and cell replacement. There are other paracrine, autocrine effects, fibrosis inhibition, GT2M activation of host tubuli. And this is something, again, this has been published, so I'm not going to go into details, but it's not only about human renal epithelial cell engraftment. It has more to it. What about GFR? What about creatinine clearance? This is a study we did on AAT NOD skin mice, and you can see that the trend of treated mice in terms of blood creatinine and GFR is leaning towards the treated mice. This is statistically significant. I want to mention that we didn't see an effect after the first injection. And again, you know, regenerative medicine is not, it's like a drug. I mean, to think that it would just, one shot would be enough is sort of naive. And we sort of saw the trend after 3 transplantations of human kidney spheroids in mice. And you can see that actually the delta creatinine clearance, there's less diminishing in blood creatinine in treated mice, and there's more elevation or less diminishing of GFR in untreated. So it's something that is being you know, this is the main issue. Can we really elevate creatinine clearance? Can we reduce blood creatinine? And again, the results show that only after repeated cycles of cell therapy, you can see the repeated measures model leaning towards statistically significant result. It's not easy. What about blood vessels? I mean, I showed you how you can generate epithelial cells, tubular segments, and this is the vascular network in the kidney. And the kidney needs oxygen. Of course, chronic kidney disease has this, you know, vessels are really sick. There's a definite need to actually, I would say, make these kidney breathe, or I call this the kidney needs oxygen. And here we don't need human kidney stem cells. To generate vessels, you don't need human kidney stem cells. You can take 2 types of stem cells. You take them from the bone marrow. You can take mesenchymal cells to generate the parasite, and you take cord blood cells, which harbors endothelial progenitors, and you mix them. You inject them into the kidneys, and very nicely, you can see formation of blood vessels. You see that the mesenchymal cell, or the mesenchymal stromal cells, generates the parasite, and the endothelial progenitor generates the endothelial cells. So here you would need to go to a different type of stem cells to generate the blood vessels. And we like to view this as a mix for regeneration. You know, you inject 3 types of stem cells, okay? You form the kidney tubules with the, you can take the human fetal kidney progenitors, but if you want to make it autologous, of course, you go to the adult and you generate kidney spheroids and you transplant them together with EPCs, endothelial progenitors, and mesenchymal stromal cells. And then you have blood vessels with blood supply and renal tubules. And this is the generation of what we call renovascular units from combined cell sources. this is how it looks. These are, this is subcapsular implantation of EPCs and MSCs, right? Endothelial progenitors with mesenchymal cells. You can see the generation of these capillaries, and these are mouse blood vessels. So it's interestingly, the blood vessels are very easy to hook up with the host vasculature. I mean, the donor vessels, it's much easier to connect blood vessels as opposed to renal tubes, which, as I showed you, we're not still sure that are connected to the nephron. And again, these are renal tubes, and these are peritubular capillaries. These are all human cells. This is, again, the 5-6 nephrectomy model, intraparenchymal implantation of 3 types of stem cells: the kidney spheroids, or the kidney forming cells, the EPCs, MSCs, and you can start seeing here, you see these peritubular capillaries and these epithelial structures which are renal tubes. You can generate even larger vessels, SMA, CD31 plus, larger vessels, and this is how it looks. This is the generation of renovascular units from combined cell sources. So we have, as I showed you, by now, the grafted blood vessels, actually anastomosis with the host vasculature. The grafted tubule, we're not sure. I mean, we have some evidence, I'm not sure it's strong enough. And this is the recipient. So we're not generating glomeruli, but we do see a functional beneficial effect at the creatinine clearance level without generation of glomeruli. And we think that it's by, enlarging in the nephron mass in case of connection and or antifibrotic effects of engrafted tubules. I mean, engrafted tubules, as opposed to mesenchymal stem cells, for instance, other forms of cell therapy can really stay for a long time. Once they generate the structure, they have this morphogenesis, they can stay for a long term in the host kidney. So they can really impose a paracrine effect for a prolonged time once they generate the grafted tubule. But this has just been published, and there's a nice editorial that really shows what means as of now the renovascular unit from combined cell sources, and this could be your own cells, of course, adult MSCs, adult EPCs for vessels, kidneys, your own kidney spheroids for kidney regeneration. So this is just the last slide, the extraction of adult kidney cells, as I showed you before. But we don't need to use adult kidney cells. We can use urine. And I'm not going to show you the data. We're pushing towards using adult urine for what I like to call it as recycling. As I told you, we're losing kidney cells. We have tremendous culture systems now for culturing urine, expanding, generating kidney spheroids from urine. And this is just a non-invasive treatment. In at least the extraction part. The implantation part would be more difficult. At least when you change a biopsy of the adult kidney to urine, makes your life much more easy. And that's just the vision of using urine to recycle your own cells for generating the kidney spheroids for your own kidney regeneration. I'm going to finish. This is the supporting that we have received over the many years of pursuing kidney regeneration, and Israeli funding is amazing. And there are other granting agencies towards that we've been able to receive, and of course the team. I like to call this the kidney repair shop. The team at Sheba, wonderful individuals, and of course. They contain the next generation of what we call Israeli Academy of Scientific Medicine candidates, and they're tremendous. So I'm open to questions, and I'm finished.
Malka, you're muted.
That there's a question on the chat from Shai Israeli. Can you see that? And I know that Carl sent you a question as well.
Just a minute.
Can you see the question, or would you prefer that I read it to you?
Shall I ask? Do you hear me now?
Yeah, you can ask, sure.
Benny, this is, this great and visionary. Now, your vision is that there is a kid or an adult with advanced renal failure, and then you'll take his or her urine, you grow spheroids, you inject them to the kidney, etc. So have you looked at the potential of cells that are secreted in the urine from patients with actually renal failure?
Well, it's just been submitted. I hope you get the. But I'm too bad we're not in the same field, Shai. So this is the the vision. It would be to use urine cells, of course. From advanced renal failure, it's interestingly, luckily we can get a lot of patients from both, I would say, glomerular causes of advanced renal disease and tubular causes. And of course, tubular diseases have an advantage, right? Because they spill more cells. And interestingly, when you generate the spheroids, you can really, for instance, we have Fanconi disease or different other types of disease. You see that the kidney spheroids can actually, you see that rejuvenation, or you see that the cells that actually are selected in culture, can reactivate pathways that are retarded in CKD. For instance, fatty acid oxidations, fatty acid oxidation stops in CKD tubules, and you can see them. You can see it reorganized or reactivated in the nephron spheroids from the urine. But the goal in my opinion, Shai, is to really understand a lot of technical issues. We have a huge issue of engraftment in the host kidneys. These are not blood cells, and when you look at satellite cells that have been transplanted into muscles or other groups and other, the problem is how to really improve engraftment. And the cell sources are fantastic. The cells are generating and they're expanding, but currently one is not the urine is great, but how in a way we can really expand the enhance the engraftment potential in the kidney tissue. I mean, luckily you're working with blood, so blood stem cell transplantation is amazing, because the blood flows, and you can see the stem cells in every part of the body, in the niches. And here the niches are much harder.
But what you showed us in mice, sorry, I'm just stealing another. What you showed us in mice show that these cells potentially are best for generating tubuli.
Yeah, right. Yeah, but remember, you look at the work of Kathleen Sustak, you know what determines GFR, the proximal tube. I mean, if you can replace the proximal tube, you might be able to I would say, get the renal function enhanced, but there may be much more to, to these engrafted tubules than, increasing the nephron mass. And we might not, because generating the whole kidney, we've transplanted fetal kidneys, embryonic rudiments. They had tons of glomeruli grown in these fetal kidney transplants, but still the GFR was mere. So maybe engrafted tubules in a way can do the job without glomerular structures, but this has to be seen in the patients, not in mice, of course.
All right, Benny, there's a question from Ehud Barniv. Do the generated glomeruli contain all relevant cell types but fall a little short in their architecture, or are there cell types missing or significantly depleted?
So you can generate glomeruli from pluripotent stem cells in the petri dish. These are immature glomeruli, and you can really, even when you transplant these glomeruli, they get blood vessels from the host growing into these glomeruli. So you can generate, from the adult kidney spheroids have no way to generate glomeruli, okay? These are just tubular network, and that is being reorganized or self-organized after we transplant the cells. So the adult cells don't generate glomeruli. only the iPS kidney organoids can have glomeruli. And again, these are immature glomeruli. So if you want glomeruli, we still need to wait. We still need to wait to get, I don't know, a much more robust system to generate more glomeruli. We're trying to sort of bioprint a tubule to maybe, stimulate secretion, just have the pouch of tubules and stimulate secretion. If you have hyperkalemia or metabolic acidosis, this might, a secretion pouch might be enough without filtration. I'm not sure. But at the moment, we have blood vessels connecting and these engrafted tubules, which are very, they self-organize, so we don't have glomeruli after transplantation. I have to emphasize that.
Okay, last question. Carl, would you like to ask?
Yeah, sure. Benny, that was fantastic, of course. And I have a few questions which I'm going to skip over because we can talk about them later, which are in chat to you. I'll just, as you're now answering Shai and others, I was thinking, you mentioned a few times how the tubule, without generating new glomeruli, can improve GFR. How can that be? And what about tubular glomerular feedback? In other words, have you looked to see whether the supplemental cells, paracrine effects thereof, are feeding back in the classic age-old tubular glomerular feedback model, which modulates single nephron glomerular filtration rate?
Yeah, that's a great question, of course, Carl. I just, we didn't do that. We did, you can generate a 2D culture and sort of co-culture them with fibroblast or generate a 3D culture with fibroblast. And you can see, even from CKD patients, right? And you can see very nice dampening of collagen production, fibronectin production, these pro-fibrotic molecules that are all over the place in CKD. So there's no question about it that you can really dampen the pro-fibrotic response by generating epithelial segments on, close by to a fibrotic tissue which develops in CKD. So that might be an explanation. You reduce fibrosis, of course, GFR is elevated. So tubular glomerular feedback is, of course, a great comment, and we haven't looked into it just specifically into the anti-fibrotic effect. And here the spheroids have, again, an advantage over other cell sources because they stay for a long time in the tissue. As opposed to, for instance, an MSC, which, the cells, which I say this, cells that don't generate the structures, cells that don't connect one to each other don't stay. You need a buddy. You need someone to help you out after you're engrafted. If you're not part of a structure, we don't see, 2 months after transplantation to mice, we don't see individual cells survive in the kidneys. You just see the structures and the cells that coalesce one to each other, or they, combine with the host. So this is, I think a lesson, for the academy. We need to stay together, or anyone, any group. The cells that stay alone don't survive.
Thank you, Benny. Rivka?
Well, thank you so much, Benny. That was really an incredible presentation. By the way, I do like your lab logo. It is fantastic. All right, this concludes this session. Wonderful session as far as I'm concerned. And we're all looking forward for the second presentation on this series. So thank you very much for all participants and have a very good day or night. And see you all.
Thank you.
אז אתה לא מוכן